Hi there, welcome about carbohydrates. So this is a much bigger spec point than the last few. So the first thing is knowing the difference between the different type of carbohydrate molecules. So let's go through those first of all. So the names are quite self-explanatory. So we've got mono, which means one. We've got di, which means two. And we've got poly, which means many. Okay, change it to whatever you want. So our types of carbohydrate, kind of aligned with that, is we've got monosaccharides. Okay, which means that they're made of one sugar unit. We've got disaccharides, which are made of two sugar units, and we've got polysaccharides, which are made of many sugar units. Okay, now the sugar unit, the most basic form, is a glucose molecule. Okay, so often we talk about glucose molecules because that's the one that you need to be able to draw, but you need to be aware that there are other types of sugar units as well. There are other monosaccharides. But it's just that alpha glucose is the one that you need to be able to draw, which is why we talk about that one the most. Okay, so they're the names of our carbohydrate molecules. Monosaccharides are made of one sugar unit, disaccharides are made of two, and polysaccharides are made of many. So if we were to draw the simple form of them, the monosaccharide is going to look something like, oops, sorry, right there. So the monosaccharide is going to look something like this. Okay, the disaccharide is going to look like two of those joined together, and the polysaccharide is going to look like loads of those joined together. We don't draw a lot because it takes us all day, but you get the idea. Okay, so one, two, and many. Monosaccharide, disaccharide, polysaccharide. Now, these polysaccharides often aren't just a single chain. They can either be a single chain that's kind of curled up into a helix like a spiral, or they can be branched. Okay, so they do vary a little bit. So let's have a look first of all at glucose. So glucose is a monosaccharide that you need to know about. So monosaccharide glucose. It's actually alpha glucose. There's two types of glucose, alpha glucose and beta glucose, and the one that you need to be able to draw is alpha glucose, but you do need to know the difference between alpha and beta as well. So let's start off with alpha glucose. So glucose has 6 carbons, so it's a 6-carbon sugar. So it's got 6. It's when I press the button on my pen, it does that, I just realised. So it's got 6 carbons, and that's really important. So we can sometimes call it a hexose sugar because it's made of 6 carbons. It contains 6 carbons. And we need to know that there's 2 types of glucose, alpha glucose and beta glucose. And the symbols for those, alpha, it's like that, which we're going to draw. And remember that there's also a beta glucose, like a capital B, but with this tail down there. So we're going to look at alpha glucose first of all. Now you need to know how to draw this. So the first thing is the top right-hand corner is an oxygen molecule. Now I suggest you draw along with me on a piece of paper to learn how to draw this. So we draw an oxygen in the top right-hand corner, and then we draw a shape with 6 sides. Okay. On this top right-hand corner, we've got an oxygen, and on all the others, we've got a carbon. So fill those in. All right. Now get a new piece of paper or fold your paper over and do that again from memory. So oxygen in the top right-hand corner, six-sided shape. All the other corners are carbon. Next. This oxygen doesn't need any more bonds drawing from it, but the carbons do. So all the other carbons need two more bonds. So one up, one down, one up, one down, one up, one down, one up, one down, one up, one down. Okay? The next thing to know is that these two here are different. So I'm going to just draw a yellow circle around those just to remind us that those two are different. I'm not going to fill those ones in yet. But all the others have hydrogen at the top and a hydroxide at the bottom. Hydroxide is hydrogen and oxygen. It's drawn OH. Hydrogen at the top, OH at the bottom. Hydrogen at the top, OH at the bottom. Okay, so let's go again. We need to just practice. So, what goes in the top right-hand corner? Oxygen. How many sides to the shape? 6. Goes on all the other corners? Carbon. How many more bonds do we need to add? 2 more to each carbon, no more to the oxygen. Which two carbons are different? So it's these two here, but I'm not going to draw that circle on there this time. I'm just going to remember that those two are different. What do we need to put on the other ones? Just do the different colour so it doesn't get so confusing. H, OH, H, OH, H, OH. Oh, I've gone wrong. Look, those two are meant to be different. Here, H, OH. Right, those final two that we've said are different are the other way up. So the H at the bottom, OH at the top. H at the bottom, OH at the top. Now this one is actually more different than that. It's actually CH2OH. And that's our molecule finished. Let's do it again. So, starting off, top corner, oxygen. Draw the sides, six-sided shape. What goes on the other corners? Carbon. 4 bonds for all the carbons. These two are different, so leave those, but the others are H, OH, H, OH, H, OH. And this one, other way round, H at the bottom, OH at the top, H at the bottom, OH at the top. This is actually CH2OH. And that's your alpha glucose. So you need to be able to draw that from memory. You need to practice that again and again until you can draw that from memory. You must be able to draw alpha glucose. Okay. Beta glucose is slightly different in that these two are the other way around. So it's OH at the top and H at the bottom. Okay, so if we were to draw that, we rub out what we've got here. So alpha glucose is the H at the top, the OH at the bottom. If we were to draw beta glucose, carbon back in, it would be H at the bottom, OH at the top. That's the only difference. A little bit about our glucose. So first of all, solubility. So glucose is soluble. Okay, so that means it dissolves in water, which is really, really useful. And that's useful because it can be used to be transported around. So we find glucose in various places. So in plants, it can dissolve, move around the plant. As you know, the glucose is made in photosynthesis, so it can dissolve in the water, travel around the plant. Equally, it could dissolve in the water of the plasma of our blood. It could be transported around organisms, like humans, for example. Okay, so it's really useful to be soluble because it dissolves, it can be transported around. Okay. Now, each of those bonds in the glucose molecule, so if we go back to our picture, each of the bonds here, when you break those bonds, they release energy. Okay. And that's really useful because glucose is really an energy storage molecule. All right. And when we break the bonds, we release some energy. And that's the whole point, isn't it? In respiration, we break down glucose to release energy. It's because we break those bonds. The bonds are holding the energy. When we break the bonds, we release the energy. Okay. So glucose is an energy storage molecule, and the energy is released in respiration, and the bonds are broken. Okay, and when we say energy is released, we know that when we get to A level, we start talking about energy being stored in ATP molecules. So the energy is released as ATP in respiration. You could say ATP molecules. Respiration when the bonds are broken. When we join these glucose molecules together, we can then start to make other types of molecule. So if you were to join two glucose molecules together, you would make a disaccharide. And if you were to join lots of these glucose molecules together, you would make polysaccharides. Okay, so we know that two molecules together, two of these sugar units, so glucose is a sugar unit, form the disaccharide. Now, I've got a really silly way of remembering disaccharides, but it works for me and it might work for you. So, MGG, SGF. And I can't just randomly remember them, I've got silly ways to remember them. So, this one is my great grandmother, and this one is silly green frogs. Let me tell you what they stand for. So, this tells me that the disaccharide maltose is made of glucose and glucose. So two glucose molecules. This one tells me that the disaccharide sucrose is made of glucose and fructose. Now, you don't need to know how to draw the fructose, but they might draw it for you in the exam question. I'd ask you to draw the disaccharide. Okay, so they might ask you to do that. And then there is another disaccharide. I don't have a way of remembering it because I can just remember it. And the last one is called lactose. And I know that that one's made of glucose and something because they're all made of glucose and something. And galactose. I just find that easy to remember because lactose and galactose sound the same. Okay. So there are disaccharides, and when we join two monosaccharides together, we make a disaccharide. And so our disaccharides, even if they're made of fructose or galactose, they're kind of the same shape. They're always going to look like two of these kind of shapes joined together. So the maltose is two of these glucoses here, these alpha glucoses. Remember, we changed that one to beta, didn't we? So two of these alpha glucose molecules joined together would make maltose. A glucose, the one we've drawn, and fructose is just slightly different, would make sucrose, and glucose and galactose, again slightly different, would form the disaccharide lactose. So then the next stage is if we were to join together more than two of those sugar units, we would make a polysaccharide. Oops. Polysaccharides. So this means they're made of many sugar units, so more than two, probably lots and lots. So an example of this could be glycogen, and another example could be starch. I'm going to go through these two together because they're kind of equivalents. So the glycogen is how we would store starch. So it's the storage molecule to store starch in animals. So it's the storage molecule in animals. And it's basically lots of glucose molecules joined together for storage. Starch is the equivalent, still a storage molecule of lots of glucose molecules joined together. This time it's in plants. So they're equivalents, but glycogen is the storage molecule we find in animals, and starch is the storage molecule that we find in plants. Okay. If we go back to disaccharides a second, I just realised I wanted to add something else about these. So disaccharides are also soluble. They're still very small molecules, which is what makes them soluble. So monosaccharides are soluble, but disaccharides are also still soluble. So that's useful because they can also be transported. They also dissolve and can be transported around the plant or around the animal. Okay. The bonds in disaccharides contain more energy than in monosaccharides. You could release more, it just means in total we've got more energy that could be released if they're broken down in respiration, which is useful. And then let's have a look at the shape. So we've got the general shape of disaccharides there. Let's start to have a look at the general shape of our polysaccharides. So we're going to have a look at glycogen first of all. So the animal's storage. So let's put this down there a bit. And let's have a look at glycogen. So glycogen, if we're to draw it, use our red. So it's lots of alpha glucose molecules joined together in a line, like this. The next thing is where the bonds are that are joining these together. So we call these bonds here 1,4 glycosidic bonds. So all these ones here, where they're joined in a line, called 1,4 glycosidic bonds, and that's because they're joined to joining carbon number 1 to carbon number 4. Okay, so if we go back to alpha glucose molecule, this is carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, carbon number 6. And this carbon of one molecule is joined to this carbon of the next molecule, and that's why it's a 1,4 glycosidic bond. Okay, so we've got 1,4 glycosidic bonds. Now, in glycogen, we've actually got some little branches. And these little branches are called are joined with 1,6 glycosidic bonds. Now, carbon number 6 is the one that sticks up here. Okay, so if we were to have a branch, the branch is a bit tricky to draw on my picture, I haven't left a lot of space. Just have to go over the top of each other. Branch might come along like this. So shape one. Can you do that one? Okay, so here, joining in here, I don't think that's even quite, it's not quite right. Let's go back. Oops. So we want to join it. Let's draw it off the end here, it'll be easier. So this is our carbon number 1, and then we want to join to the carbon number 6. So let me draw maybe, oops. You're not going to have to draw this precisely, but just trying to make a point to explain it to you. So really, our little carbon number 6 is up here. Okay, so we got a bond from there to there. Maybe I should join this. So I've got a bond from there to there, joining that in. And then this might just continue on in its chain this way here. So you can see that I've got a branch coming off down here. And you can see where it's joined with that little blue line there. And that bond there, that one, is a 1,6 glycosidic bond. Okay, can you see that? So these are 1,4, that's going to be a 1,4 just there. It's going to be 1,4 just there. So wherever it's a chain, equally that there is going to be a 1,4 just there. This one here is going to be a 1,4. But where the join meets for a branch, that's a 1,6. So you need to be specific that glycogen contains 1,4, 1,6 glycosidic bonds because it's branched. Okay, and that is really, really important. So because we've got these branches in the glycogen, oh, where's the D gone off the end of there? Because we've got these branches on glycogen, we've got access to break off a glucose molecule in more places than if it was a straight chain. So if we had a straight chain molecule, or coiled or any shape, but one chain, we could remove this glucose here, we could remove this glucose here. And in breaking these glycosidic bonds here, and then respiring the glucose molecules, we could release energy. Okay, now we only got 2 glucoses accessible. Now, as soon as we've got a branch, we've then got 3 glucose molecules accessible. If we had another branch, we've then got 4 ends. So we've got 4 glucose molecules accessible. And that allows us to have a quicker access, quicker supply of energy. So that's why it's really, really useful. So the fact that it's branched means we've got more access to glucose molecules, because these ones in the middle are kind of trapped, aren't they? To glucose molecules to break those glycosidic bonds. Okay, and that means that we've got a quicker supply of energy. Because obviously glucose molecules are respired, which releases ATP molecules, which is our energy. Quicker energy supply. Okay, the next thing about glycogen, so we've got that it's branched. That's kind of our key thing. That's what that picture's showing. That's what our bonds that we need to quote are. That's what our explanation there is kind of saying the benefits of. The next thing about it is it's compact. Okay, so the reason why it's useful that it's a compact molecule, that means it doesn't take up too much space in our cells. The reason why that's useful is because we can store lots of it in a given space. Okay, so we can store lots of glucose molecules by making them into glycogen molecules for storage. They come nice and compact, they don't take up too much space. We can store lots in a small space. The next thing is that glycogen is not soluble. Okay, or you could say it's insoluble. It's not soluble. So we said that our monosaccharides and disaccharides were soluble because they're nice and small. Glycogen is actually quite a big molecule, so it's not soluble. And actually that's useful when it's being stored because glycogen is stored in both your muscles and your liver. Okay, and that means it actually stays where it's stored. It doesn't dissolve in the blood and move off around the body. It stays where you put it. It stays in this where you want it stored in the liver and the muscle cells. So that's really useful. Another description of it is you could say it's large. It's a large molecule because it's made of lots and lots of glucose molecules. And because it's large, it's made of lots of glucose molecules, it's obviously storing lots of energy. Okay, so that means it's a large energy store. So it's a large molecule, which means that it's a large energy store. It stores lots of glucose molecules and therefore the potential to release lots of energy. Okay. Next, we're going to go on to starch. So remember, starch is the equivalent to glycogen. Glycogen is our storage molecule in animals, and starch is our storage molecule in plants. So let's write it on here because obviously we've run out of space on this one. Oops. So this time let's look at starch. So starch is our energy storage molecule for plants. And let's have a look at the structure of it. So glycogen was made of these glucose molecules in a chain with branches. Okay. Now starch can come in two types. All right, so we've got two different polysaccharides that make up starch. The first one is called amylose, and the second one is called amylopectin. Okay, so let's add on to here. And it's made from two types of poly. Right, so our two types are amylose and amylopectin. So the amylose in the simplest form, simplest drawing, oops, looks like this. And amylopectin is like this with branches. Okay, if we're going to draw that kind of simple diagram for glycogen, we would draw it as a straight line with branches. Okay, so what this really is a chain of glucose molecules like we drew before. Okay, but twisted, spiraled up into a helix shape. Okay, it does not have any branches, no branches. This means that it increases the amount that can be stored, so you can store lots in a small space. Okay, so increases amount stored in small space. Okay, so there's no branches. It's got this spiral, this helix, we call it, like the double helix of DNA. It's a single spiral. Because it's coiled, it's very, very compact. Again, increases the amount that can be stored in the small space. Now, amylopectin is different because it has the branches on it. All right, so it's still got the spiral shape, okay, but this time it has branches. Now, remember back to the type of glycosidic bond. When there's no branches, all the bonds are going to be 1,4 glycosidic bonds. Here, because we've got branches, it's just the same. It's going to be 1,4 on the straight sections, and 1,6 where you've got a join glycosidic bonds. Okay, so make sure you know the type of glycosidic bonds that are involved. Remember that because we've got branches, it's really useful because we've got quicker access to the glucose quicker access to that energy. Because we've got more ends. So rather than this one, we can only kind of release this glucose and this glucose for respiration. On this diagram, we could release this glucose, this glucose, this glucose, and this glucose. So we've got access to more glucose molecules, more ends. That's the advantage of having the branches. Okay. A couple more things about the starch. As a whole, starch is not going to be soluble because, again, it's a really big molecule. It's not soluble, it's insoluble. Again, makes it a really good storage molecule, just like glycogen. It stays where you put it, and the starch is stored in starch grains, and you might remember that they're called amyloplasts. Okay, so whereas our glycogen was stored in muscles and liver, our starch is stored in the amyloplasts, which are basically like starch grains in the plant cells. Remember, they're in plants, aren't they? Okay, so it makes it really good for storage. Starch molecules are really large molecules, which means that they contain lots of energy. Okay, lots of glucose, and therefore lots of potential for releasing energy as ATP molecules. Alright, so let's just make sure that they're obvious, soluble and large. Okay, so that brings us to the end of that spec point. It is a long spec point, there's a lot to do there. So you need to know, first of all, I think how much we've done. Monosaccharides, disaccharides, polysaccharides, and what they mean. 1 sugar unit, 2, and then many. So our monosaccharide that we need to know is alpha glucose, and then we taught you how to draw it, so make sure you can go through that and draw it. Like that. The difference between beta is these two here were swapped over. So alpha, the hydrogen was at the top. Beta, the hydrogen's at the bottom. We talked a little about the properties of glucose, what the names of the disaccharides are, and the properties of the disaccharides, what the polysaccharides that we need to know are, and then the structure and properties of glycogen and the structure of properties of starch. And remember, glycogen is your storage in animals, and starch is your storage in plants.